Okay, we've been through translation. We've talked about all the components. So I thought maybe just to take a very few minutes to talk about some of the ways you might want to study for that and review, as well as just a couple of problems that kind of traditionally show up and are asked about translation type things. So first, what do we need to study? Well, remember all the process of translation from mature message to making that polypeptide. That's where we're going. Think about all the steps. Think about tRNA charging, how all that occurs, formation of the ribosome, what a ribosome is, as then, and the initiation process, elongation, and termination. When and where? Well, it happens, as we said, primarily in G1, also some in G2 for most cells. Where in eukaryotic systems it occurs in the cytoplasm. What are the components, the elements we need? There are these units we talked about, obviously the message, the ribosome formation, tRNAs, and of course you need a coding dictionary in order to do it. Steps are activation, initiation, elongation, and termination, at least from our perspective. You may see them called other things, but they're basically 4 steps, sometimes just 3, with tRNA charging kind of being off on its own, not included as a step. But we think of it as important enough that it's critical to the operation, so we call it a step. Right? So here's a couple of little simple problems, okay, and just to give you some idea. So you find out that there's a polypeptide that's got 100 amino acids. What's the number of codons that were required to produce that? Well, that's just a backwards way of asking you, what's the relationship between a codon and a amino acid? Is 1 codon equals 1 amino acid. So you've got 100 amino acids, so somewhere around 100 codons were required, okay? And it could be any number of that. Now the question mark here, you need to be careful. Some people say, well, you've also got to include the stop codon, even though there's not it's not a stop. There's no amino acid there. That's how many would have been there, but I'm not going to work that here. Anticodons, 1 anticodon for each codon, right? So 100 would be a good answer for that one. Number of tRNAs needed to do this particular translation, that would be here, would be 1 codon equals 1 anticodon equal 1 tRNA for our purposes. Okay, so each tRNA would have to bring in one of those amino acids, however it was constructed. So we would need 100 tRNA visits, right? And it depends on whether, different tRNA is a different question, because it depends on what the amino acids are. If you had a string of serines, that would have all been the same tRNA anticodons that are there. But for our purposes, just think of it as 100 unique amino acids. Then this question is a little tricky, this number of nucleotides needed. It's almost always considered to be the number of codons plus 3 nucleotides is the minimum answer. By that I mean you need 1 codon for each of the amino acids, so you need 300. You need 300 bases, right? 300 nucleotides. Because you got 3 for each amino acid, and then you need a term code. That's where that term code back up here comes in. Okay, so I would say the answer here would be something like 303, might be a good answer. All right, range. Sometimes you see the answers in some books as 304 because they count the G cap as critical for translation, because that cap is what really attracts the whole unit together and what pulls in. And remember, kind of a hook-like structure with that G cap actually acts there. So some people include that. So sometimes you see plus 3. So you see the number of codons times 3 plus 3 plus another one. So you might see, plus 4 here. Okay. But you start out initially just thinking, okay, a number of codons times 3 nucleotides. I need 300 minimum. Okay. I need a stop codon. That's another 3 that I've got to add in. Okay. Nucleotide, so it's 303 in this case. And then if I want to throw in that GCAP, but the reality is these messages, are quite long, and you don't translate every piece of it anyway. So it's, the modern world that varies, but in most general textbooks that'll work for you. And then there's an interesting question. Would this be the likely number of nucleotides present in a transcriptional unit if you're in a mammalian gene? Okay. In other words, whether you come up with 300 or you come up with, with 5,003 or whatever the number is that length of that message what you would see in the mammalian gene? And the simplest answer, the answer to that is no. The simplest reason for why is that on the very least in mammalian genes always have introns. So remember that loopy thing, that picture of loops that we had? uh, where we had complexed up the message, the mature message with the original DNA, and it looped out sometimes. We called those introns. So no, the actual transcriptional unit that you originally took that message from is often much larger, and sometimes, twice as large, if not more, than the actual mature message that you end up with because you splice out all of those introns. So the answer is no for that. If you move up the ladder and talk about in biochemistry, and you talk about in cell and molecular biology, there's another no, and the other no is, besides removing the introns, which is a simple answer, you've got those non-transcribed regions, right? You've got those poly A tail areas on the 3'. On the 5', you've got a fairly long area there in some messages that's not going to be translated, even though it's transcribed. So even that, so it's never going to be directly equal to the number of nucleotides that produce the amino acids. It's always going to be a bigger message required to get it done, at least in eukaryotic systems, which this is why the word mammalian gene is there to focus you on that you are in an advanced system in a eukaryotic environment. Okay. All right, let's go backwards the other way. Here's a sequence, and this is an amino acid sequence, and they want you to tell me about the nucleotide sequence that would have produced that, right? So first question, is there a start codon? Where do you start looking? You always start looking at 5'. Remember, the 5' end is the one work from, okay? And the answer here is yes, there it is right there, okay? Now, it could have been much longer, instead of just 1G, we could have had lots of bases out here, but we could have had that, okay? Now be sure to look for the 5' in and make sure you understand what you're doing, because if it were labeled 5' over here and this 3', you'd have to work from this direction, okay? And you keep working along and working along and working along and working along. Oops, there's an AUG right there, right? AUG. If you consider this the 5' in, you'd be reading it in the other direction. So don't get just focus, in English, in English language and Latin language, we read from left to right as a straight line, and you get so focused on doing that you always read this way. But you got to look, inside of a cell, it doesn't know left from right, so it reads from 5' in. So be sure that you pick the right end. We picked the right end. We found that there's this AUG, and we've got it. Okay. All right. All right. So how many codons? Well, remember, once we have this small subunit in place and we frame this first start codon, then we just frame 3 letters after that. It's commaless and it's straightforward. So we just mark out every 3 and count them up, okay? So in our case, there's 1, 2, 3, 4, 5, 6, 7, 8, it looks like, codons. All right, is there a stop codon in here? Well, we're looking down here somewhere for UGA or UGA or something. No. All right, when that question is there, it makes you have learned what the term codes are, right? So you need to know what the term codes are and go look for them in here. And that'll play an important role when we do things like we do some mutations, right? If you were to drop out this U right here, okay, then in fact you might well have a term code in there somewhere that's not present right now, okay? Or if you added in an extra nucleotide. So knowing these is important. It's a way to make you go back to that the rules of the dictionary and think about what term codes are. Right? What's the anticodon sequence? In other words, what would you find in the tRNAs? Well, you just find the opposites of these, right? So A would give you a U, right? Because remember, we're using uracil instead of thymine because it's RNA. A U would give you an A G would give you a C, and you just keep writing. That's exactly what we've done here, right? How many amino acids would be added? Well, 1 for each codon. There's a 1-to-1 relationship. And what would they be? Well, in our case, valine, leucine, threonine, met, leucine, valine, phenylalanine would all be added. How would you know that? You'd have a coding dictionary. Would we give you a coding dictionary on test? Absolutely. Okay, we're not going to expect you to memorize all 64 of those amino acids relative to the codons. Why would you waste your time doing that? Just learn how to use some version of a coding dictionary, and then you'll be able to do the translation for yourself. Okay. All right, we finished translation. We've made polypeptides. We've talked about these, but we still don't have a protein. Okay, remember that there's no direct link between a message and immediately producing a protein by the translation process. So now we've got to take the string of polypeptides, string of amino acids, and make them into proteins. And that's what we're going to do in the next set of lectures.